To thank the organizers for this opportunity to come and chat with you and give a very different perspective. So I'm a material scientist, and I'm very inspired by biology, but what intrigues me is the possibility of looking at biology and trying to mimic that behavior in synthetic materials. So I'll tell you a couple of stories today, and the people who really did the work are Pratyush Dayal, who is now a faculty member at one of the IITs, Deb Debrada, and Olga Kuksanok. So there's been a proliferation of animal pictures in these talks. And I'll show you one of my favorite, and it's called the mimic octopus. And it's very, very hard to believe, but all of these images are one animal. So the way the mimic octopus evades its enemy or its predator is as it can change its shape. And it changes its shape to assume these different morphologies when it's in the presence of a predator. So this amazing shape-changing ability is vital to its survival. So we're very inspired by this mimic octopus, and I'll try to tell you a story where we try to create a synthetic material that can also undergo these dramatic shape changes. And then the second story that I'll tell you about is inspired by the amoeba. This is the second talk, I believe, that refers to the sex life of the amoebas. You know, in an alternative universe, I wonder if there are these little amoebas sitting around going, whoa, look at the sex life of human beings. But to us, anyway, they truly are amazing. And what these animals do as part of their reproductive cycle, they send a signal to each other, and as a consequence of the signal, they come together and aggregate. I'll stop there because it goes on to do very, it becomes X-rated, and my mother is going to be watching this. And just to remind you that, you know, what questions you ask, be careful because she'll be watching. So the amoeba is really amazing because not only does it send the signal, but it understands the signal. So it emits and sends a signal. So it undergoes this auto-chemotactic because it generates the chemical gradient to which it responds. And so that's also an intriguing question for material scientists. Can I design a material that emits a signal and understands that signal, and then they all come together to mimic the amoeba? And might that have a particular functionality that could be useful? So I'll tell you a story of these casts of characters. All my stories have to do with gels. Gels are mutable, deformable. So if you're thinking about creating biomimetic materials, they encompass that flexibility that allows you to do things like shape changing. So I'll tell you a story about a hydrogel that is responsive to light, and it undergoes the shape changes, and it also can undergo directed motion in response to light. And then I'll tell you an incredibly intriguing story where the synthetic material comes together to do auto-chemotactic. And that's the story that we had actually published on. But then when we looked a little more carefully, it actually did this beautiful dance where it undergoes this chiral motion. It doesn't form a chiral object, but it undergoes this chiral motion. So it forms something like a pinwheel, and it does it all by itself. And if I have time, then I'll tell you a story where we can take the stimuli responsive gel and then actually use it for a practical application. And that's, it maintains a temperature, so it performs a biomimetic homeostatic function of maintaining temperature. So let me start with the first story, and this is the one that's inspired by the mimic octopus. And you know, at a chemistry conference, people blanch at equations, and at a physics conference, I'm sure there's blanching at chemistry, and I am not a chemist, so I'll keep the chemistry to the minimum, which is all I understand anyway. So these are a particular type of gels. They're polynipam, but they contain a very lovely group, and it's called a spirobenzopyran. And Oleg's looking at me like, oh yes, I know the spirobenzopyrans. And the really lovely thing about spirobenzopyran is that when you shine light on them, then one of the chromophores goes from an open ring configuration. So in the absence of light, it looks like this. It's lovely and it's extended. And then, but when you shine it with light, then the open ring closes, and this thing collapses. So you can think of this in the absence of light as being hydrophilic, if you like, and in the presence of light being hydrophobic. And so there's this lovely group that has exploited this property to a very nice advantage. If you use a photomask, then you can shine light through the photomask and make these very beautiful relief patterns. And that's what they've done here. So the raised part is the part that's not irradiated, and the depressed part is the part that's irradiated. And the really beautiful thing is, if you remove the light, it goes back to its original shape. So it's rewritable. And you can do this remotely. You can just shine light, you get a pattern, you remove the light, it goes back to its original shape. So you can do this multiple times with the same material. So I'll show you that it's a very, very general design concept. I'll show you that when we can shine light on it through different shaped dyes, that it can undergo this photomorphing. But what has not been observed experimentally is that I can use this photomorphing to make a motile object. So how would I create a model for this? Well, we build on Flory-Huggins. And again, I'm not all that familiar with the background of the audience. I'm sure there might be some questions. But I'm going to keep it simple. Flory-Huggins theory for polymer gels, well developed. So there's an elastic energy term, C naught as the cross-link density. These are invariants of the strain tensor. There is an interaction energy, well-known Flory-Huggins interaction energy for a polymer and a solvent. And so these are the classic Flory-Huggins terms. Now we have to have a term here that we introduced. This is the concentration of chromophores, and this is the strength of the interaction, and this is effectively showing that there's a collapse due to the presence of the chromophores. There's going to be a decrease in solvation. From the energy then, I can get an osmotic pressure, I can get an elastic pressure, and I have a constitutive equation that relates the stresses to the strains. And so those are my equation. Now I have to have dynamic equations for this. So this is just a continuity equation for the polymer. Vp is the velocity of the polymer. So here's where I say there are no dynamics in my story. Okay, I'll show you that these things move so slowly. They're on the order of hundredths or tenths of microns per second. So there's no dynamics in my story. This is a Bayes relaxational dynamics, so we're in the overdamp limit. So I'm going to say that the force is proportional to the velocity. And from these equations, then, I can get the equation for the velocity of the polymer. And now if I can solve for the velocity of the polymer, then I can update my evolution equation. And my brilliant postdocs developed a method that we call the gel lattice spring model. So we actually take these equations and solve them numerically on a lattice using finite difference and finite element methods. So in addition to those equations, I have an equation for the interconversion of the chromophores. So this is a rate constant, forward rate constant. Sorry, I'm turning my back to you. It depends on the intensity of light. And this is a reverse rate constant. And I can look at the photostationary state, so I can just set that equal to 0. And the reason I just did that is just to show you that I'm going to use numbers. These kind of numbers are known experimentally, and they're known from this group. So I have an idea of the order of magnitude difference between the forward and the reverse rate constants, and roughly what the steady state concentration for those chromophores is, and so I can take those numbers. What I'm going to try to tell you is that there's only one adjustable parameter in my model, and that's that alpha that characterizes the strength of the interaction. All the other parameters I can take from experiment. And so I can calculate the equilibrium degree of swelling. I can do that analytically, or Olga can do that analytically. And so I can set the osmotic pressure in that circumstance that the osmotic pressure is equal to this elastic pressure. And I can get the equilibrium degree of swelling in the, relative to, in the presence of light relative to the, when it's illuminated. And here it is in the absence of light. These dots are the simulations. The line is this analytical calculation for in equilibrium. And when I increase the intensity of light, you can see that the equilibrium degree of swelling decreases. It shrinks, just as I told you. And this transition with temperature becomes more gentle. Also, this delta lambda, which is the peak in relative shrinking, so it's the lambda dark minus lambda as a function of the intensity, has a peak at a certain temperature. Now here's what we see.
This has temperature dependence because there's temperature dependence in the parameters in the model?
Yes, there's a temperature dependence in the Flory-Huggins chi-parameter, and that temperature dependence I know from experiment.
Okay, so it's a temperature dependence that comes from polymer data.
Yes, it comes from polymer. Okay, and then, so this is data from experiment. Here's the lambda for the equilibrium degree of swelling in the absence of light. Here it is the presence of light. So we see the same qualitative trend, and then we see the same qualitative trend for this delta lambda as well in terms of here's our calculation, and here's the experiments. So that's a corroboration of the model. But here's now the fun part. So we take that model and we say, let us have an aperture. And let's have the aperture, we can control the shape of the aperture, and we're going to shine light through the aperture. And so with this one sample, and this sample is on the millimeter size scale, I can make all these different shapes just by where I shine the light. I think blue here is the part that is illuminated, so that's the collapsed part, and green is the swollen part. So this one sample gives me all these different morphologies simply by where I choose to shine the light. Okay. All right, and so you can generate a zoology of morphologies. It's partly limited by imagination because these apertures are relatively easy to design. So,
how quickly does it work?
Oh, you shouldn't have asked that. So it's about an hour, okay, or hours if you push me. But this is millimeter size pieces. So that is a small disadvantage. But to defend my little beautiful sample, it's taking hours, it's rewritable. Yeah? So that's a really, really nice thing about it. Okay, so what can I do with this? I mean, I showed you that beautiful relief pattern, so people have known that you can inscribe a pattern onto this. So this is what's not been observed experimentally. What if I take that light and I raster over the sample multiple times? Then this is what happens. Now recall that there's no hydrodynamics. Partly hydrodynamics doesn't play a very big role. These are moving so slowly, on the order of tenths to hundredths of microns per second. But what's happening, and I should have pointed out more carefully, but I think I did, that the dynamics comes through the interdiffusion of polymer and solvent. So if the solvent is pushed one way, the polymer will move the other. And that's what's happening here. As you swipe the light, then I'm progressively pushing or squeezing solvent in that direction, and so the polymer moves in the opposite direction. Okay? And one little step for this guy looks, you can see the movie's a little bit better here, looks like this. You take a little step to the right, but it undergoes a larger step to the left. So it moves in opposite to the direction to which I'm moving the light. I'm moving the light, I'm pumping out solvent this way, so the polymer moves that way because of my incompressibility constraint. Remember, so the only motion is the interdiffusion of polymer and solvent. And so that's one little step. And so then after multiple swipes, this happens to be, I think, 30 swipes, this undergoes macroscopic motion. Again, it's small, it's slow, but it undergoes macroscopic motion. And this phenomenon is really very robust. I can take funny shaped objects. This just happens to be a cylinder. And I can torture the little cylinder even further. I can have variations in its cross-link density. So we just did that for fun because it was a different color. And then, so even a cylinder I can move. I can move different morphologies by swiping the light. And it moves opposite to the direction in which I'm swiping the light. Now, I neglected to mention, but I'll emphasize it here, and I'll just say that it's been observed experimentally. When you shine light, this is blue light, onto the sample, there's no local heating, or if there's local heating, it's very, very small. Right? So I can use temperature and light as orthogonal variables here. And one thing that we've seen is that the displacement in a swipe varies with temperature. So the velocity depends on temperature. But this plot looks very much like that delta lambda, which is a difference of the equilibrium swelling in the absence of light versus with the light. And so the peak happens in the same place. You get, so the greatest expansions, you get the greatest displacement at a particular temperature. And so there's a region where that displacement and the velocity increase with temperature. And you're probably going, oh, Anna, you're spending a lot of time on that. Why are you showing me that? And I'll show you why. Because, as I said, now I can use them as orthogonal variables. So I can have this move in a temperature gradient. So temperature is increasing this way, but I can swipe the light that way, and I can get it to do all sorts of dances. Now, the reason it's moving up this way, there's no walls. That's my simulation box. It'd be lovely if I could tell you that it was crawling up a wall, but it's not. That's just my simulation box. But it's moving upward and to my left. Is because, remember I told you the displacement was greater at a higher temperature. So if I put this little body in this temperature gradient, then the top is going to undergo a higher displacement, a greater velocity in this region. So as I'm swiping with the light this way, the top will move slightly faster as I swipe, and the net motion, and there's this beautiful complicated behavior. And I can do it, we did it with this tubular structure where we swipe the light this way and had a temperature gradient, and you can get the tubular structure to bend. Yeah?
So the light is only from one side, or it comes from
Well, no, it's only from one side. It's only from one side.
So why is it the temperature gradient? Because then the lower side shouldn't be affected at all.
Well, I want the temperature gradient because I'd like to introduce a differential in the velocity or displacement of this little piece. And with, yeah, I don't assume attenuation of the light. I do not assume attenuation of the light. Okay. So I can tailor the motion by introducing this temperature gradient. So let me finish what I have about this story. We've developed a model for this spirobenzopiran functionalized gel. We've demonstrated good agreement with prior experiments about its behavior. The light allows us to address a particular region. So Fred gave a beautiful talk about local contractions in fibrous materials. So perhaps we could create that with light, shining light in a particular region and getting a local contraction. We've also demonstrated that by rastering the light over the sample, we can get it to move. So there's another thing that you might be able to do, very novel construction. That is, you fashion a piece into a shape that you would like by illuminating it through one aperture, and then you also get it to move to a particular location. And then you get another piece to move to a particular location. And you can do construction that way. They're gels, when you bring them together, perhaps they'll stay stuck to each other. And so you can create and fashion very complicated shapes this way. So let me go on. I've told you about my shape-shifting hydrogels. Let me go on and tell you about the auto-chemotactic self-oscillating gels. And we were inspired a little bit by Igor Anderson did this beautiful, beautiful work where there are two gears here, and it's bacteria, the random motion of the bacteria, that's causing the gears to move and interact. And we were wondering if we could create something like that without the need for carrying a vial of bacteria. And then we were also very inspired by this child's toy, a pinwheel. I think we call it pinwheels. What do you call it here? What? A windmill. A windmill. All right, so the idea is, can you get a material to undergo that kind of motion through self-organization? And so the answer is yes. So I don't know how many of you know about the Balazs-Zhabotinsky reaction. Show of hands, how many of you? Okay, we're golden. So the Balazs-Zhabotinsky reaction was discovered serendipitously in the 1950s. Balazs and Zhabotinsky actually never met, but they discovered it in a petri dish or in solution, and it's a self-oscillating reaction. So it's a periodic oxidation and reduction of a catalyst, and if you watch it in a petri dish, you can see that it changes color beautifully and periodically. And in the late 1990s, Ryo Yoshida took that catalyst and anchored it to the backbone of a polymer gel. So now that catalyst is undergoing this periodic oxidation and reduction driven by the chemistry. And the catalyst is anchored to the backbone of the chain, so it cannot move away. Well, it turns out when that catalyst is in the oxidized state, it makes a more hydrating environment for the polymer gel. When the catalyst is in the reduced state, it's a less hydrating environment for the gel. So the gel swells and contracts autonomously. So as Ryo Yoshida says, and as I keep repeating, it swells and contracts. I'll go back and beats like a little heart. There you have it. All right. So there, we collaborated with Irene Chen and Kristen Van Vliet at MIT, and here's the little sample that Irene made. And it is the only existing polymer gel that swells and deswells autonomously. For everything else, you need external stimuli. You need a continuously stirred tank reactor. I bet you never thought you would hear that word in a physics conference. But you need something to regulate the swelling and deswelling. Here it's the chemistry that does it for you. You can walk away, and millimeter-sized pieces will oscillate for hours. And to replenish the oscillation, all you have to do is feed it again. You put more reagent into the petri dish, and it'll keep oscillating, so you can rejuvenate it. So the question is, can we use this beautiful beautiful material that keeps disappearing, to make self-assembled machines and self-assembled tools? And so we had developed a model for this BZ gel. We were greatly aided by the fact that there's something called the Organitor model, because it was developed in Oregon, and it was for the BZ reaction in solutions. So we could build on that, and the Organitor model was extremely powerful because even though there are dozens of components in this BZ reaction, they said, look, there are two critical variables. U, which is one compound, it's the activator, and I'll be referring to U a lot. So U represents the activator for the reaction, and V is the amount of oxidized catalyst. And they show that you could capture the behavior of the BZ reaction in solution with just those two evolution equations. And there's standard, not so standard, reaction diffusion equations. So for U, which is free to diffuse in the system, there's the diffusive part, and F is the chemical reaction part. And V, which is the oxidized catalyst, is tethered to the polymer chain, so there isn't a flux term. But G is a chemical reaction part, and this is the evolution equation for the polymer. So we modify these equations by introducing the evolution for the equation for the polymer and modifying the equations for V and U by explicitly including the volume fraction of polymer and modifying F and G, which captures the chemistry occurring, and including phi and 1 minus phi, which, remember, phi is the volume fraction of polymer and 1 minus phi is the volume fraction of solvent. And again, we have relaxational dynamics. And again, no high dynamics. The frequency at which these beat is about 100th of hertz, tenths or hundredths of hertz. So periods are on the order of minute or seconds. Okay? So again, the only dynamics is the interdiffusion here of polymer and solvent. And I solved those equations, my wonderful postdoc solved those equations numerically using, again, our gel lattice spring construct. So I'll show you a couple of slides where we try to validate this gel lattice spring model and by comparison with experiment. And a long time ago, we had said that the pattern of oscillations that you get in these gels should depend on the size and the shape of the gels. And so here from our simulations, and here's again from the simulations, and here's the experiments from Kristen Van Vliet's group, Irene Chen, the really wonderful graduate student. So here's some qualitative validation of our model. We collaborate, in addition with Kristen and Irene, with Ryo Yoshida, who again was the first person to make these BZ gels. And we convinced Ryo to make heterogeneous BZ gels so that there are patches of BZ catalyst in a neutral gel. And the point of this slide is to show you that the patches where there's the catalyst, it's in solution, it's just like that cartoon, it's in solution, they communicate with each other through the solution and through the neutral gel. So one, I want to convince you there's communication between the BZ gels, and two, I want to convince you that we actually could capture the phenomena that Ryo Yoshida saw, which is that because of the communication, there's a chemical wave that passes from one patch to another, and then there's information passed from one patch to another, and they undergo this synchrony in oscillations, and we could capture what was seen experimentally. And finally, there's another collaboration we had with Ryo Yoshida, and he said, let me take a little piece of the BZ gel, and I'm going to introduce a gradient across its width. So it's a higher gradient in cross-link density here, a lower gradient in cross-link density there. He could do that very beautifully experimentally. And I'm going to put it in solution. And then, remember, it beats like a little heart, so it oscillates. But because of this heterogeneity in the material, it bends. And if you look long enough, you'll see that it undergoes this kind of motion, and ultimately it moves upwards. And the other reason I'm putting this slide up here, because you can see very clearly a phenomena that I'm going to rely on very much in the next couple of slides, is that the chemical waves are going this way. So the waves of chemical propagation of U, the activator, and the solvent are moving this way. You can see that quite clearly. But the chemical waves move this way, which is sort of downward, and the little piece moves upward. So the motion, the direction of propagation of the piece is opposite to the direction of the chemical wave. All right, so now I've showed you our model. I've tried to indicate that we've validated aspects of the model by comparison with experiments. And so now I'm going to tell you the auto-chemotactic story. So we have not just one piece now, we have multiple pieces. Our pieces are on the millimeter size scale. And we adapted our models so that we actually have diffusion of U, the activator for the reaction, in solution. They undergo this disproportionation reaction. So we solve this equation for U in the solution. See, the period of oscillation is quite long. And we also introduce a small repulsive interaction between the gel pieces so that they don't interpenetrate, right? So they don't come into each other. There's a soft Lennard-Jones-like repulsion between the pieces when they come together. And then, now I'm going to show you magic. So think of an experiment where you would take that big piece of gel and you cut it into 4 pieces and you pull the 4 pieces apart. and you just let them sit there, then these 4 pieces will find their partners again and come back together. Right? So you do the experiment, you cut a sheet, 4 pieces, you pull them apart, and they come back together again. And they do this autonomously. So why does this trick work? All right, here's the distribution of U. So it's highest, the activator of the reaction, it's highest where it's red and lower where it's blue. So we did this experiment, simulation, where we took these little cubes, individual cubes of U, and we increased the individual cubes of the BZ gel, and we increased the amount of U in the solution. And as you increase the amount of U in the solution, it beats with a higher frequency. Okay? It's beating faster. Right? Now, we know from the collection of oscillators that the one that beats fastest is the pacemaker. It sets the directionality for wave propagation. Well, if you look carefully, my little gels were sitting at these corners. And the reason that they have such a high concentration of U in the middle is geometry. I put them over here, and so that as the gel is beating, it's generating U, the activator for the reaction. It's undergoing this chemical reaction, it's generating that U. And U is diffusing, it's diffusing into the solution. And because of the geometry, then, the highest concentration of U between 4 pieces will be in the center between the 4 pieces. And so this part of the gel will beat with the highest frequency, because it's near the highest concentration of U. It acts as the pacemaker. It sets the directionality of wave propagation. So the waves are going to propagate from this corner out. Remember I said the motion is due to the interdiffusion of polymer and solvent. So if the chemical wave goes from here to here, and again it originates from here because it feels the highest concentration of U, and that's the pacemaker. So the wave will go from here to here, then the polymer will go in the opposite direction. And because of that complicated behavior, then they come together in the center. And the behavior is very robust. I mean, I don't have to have them exactly lined in corners. I can have the corners be skewed a little bit, their initial positions be skewed a little bit, and they'll still come together and gather in the center. And so, in fact, I can measure the distance here between the centers initially for all these different configurations, and you can see that eventually they all go to a very similar value when we let them run for a long time. And that's where the story ended for us in one publication. But we didn't look carefully enough until a little bit later. If you look carefully at this one, they seem to oscillate in a beat pattern. This one, then this one, then this one, then this one. And we only noticed the consequence of that in a subsequent, current study. That's the next story I'll tell you about. Now, this auto-chemotactic, there's no experimental evidence for it yet. But there's some experimental evidence that they communicate. There was the Yoshida story I told you, that they communicate through the solution, and they communicate through that neutral gel. But there is another piece of experimental evidence that they do communicate through U. And this was this beautiful thing that Irene Chen did. And she took this, someone said smiley faces are really in, then they just had a pig, but here's a smiley face. So we have this mouth and eyes, and this was, the BZ gels are not only chemomechanically responsive, they're mechanochemically responsive. So they can be in a state where they're not oscillating, but if you press on them, then they oscillate. So if you look carefully at the mouth, she put a cover slip on the mouth, and she pressed down on the mouth with the cover slip, and you'll see that the eyes wink at you, all right? And they're not touching. So the only way they can communicate is through the solution. It's through that diffusion of U that the pressing, the mechanochemical resuscitation of the smile of the mouth causes the eyes to blink, right? And now she did another experiment where she moved the eyes a little bit further out, and in that case, the eyes don't blink. then there's that disproportionation reaction that happened to you, and they don't make it all the way to the eyes are too far apart, and so they don't blink. So I don't yet have experimental evidence for that story. I told you about the auto-chemotactic, but take this as experimental evidence for communication through you. Now, if you look very, very carefully, but I don't want to push this point, but if you look very carefully at the corner of that mouth, this one, all right? There, it popped away, and you can see it's moving a little closer to the eyeball. And you'll see that because in a second it'll pop away, all right? That's just how the initial, it's not the popping away, that's just the restarting the movie. It's the fact that the corner moved a little closer to the eye. I'm not going to push it. We don't have yet clear evidence of the signaling, but not clear evidence of the auto-chemotactic. All right, but now the story that I'm going to tell you next is even more fantastic. So I wanted to show you that, I want to at least convince you that they will communicate through you. So this is where the story ended, and we published this. And then we looked a little longer, all right? So we'll call that stage 2. And what happens in stage 2 is that they start rotating, okay? So there are a couple of necessary conditions for this rotation. They have to develop this beat pattern, and that beat pattern has to be in synchrony. So it's 1, 2, 3, 4, 1, 2, 3, 4. When that beat pattern develops, then we get this, what we call a comet of U that circles, okay? And then I get this rotational behavior. So I've made the pinwheel. And it's done it all by this dynamic self-organization. But you can think about, well, what are the mechanisms for this? There are a bunch of forces acting on these little gels. There's a force that drives them to the highest concentration of U in the middle. I showed you that. There's that generation of the chemical wave, and I told you they move in the direction opposite to the chemical wave. I showed you that with the little worm. Remember, the chemical wave went this way, and the worm moved up. So as a result of those forces, the resultant force is that they move with respect to each other in a relatively skewed manner, and then they come relatively close until the excluded volume effects kick in, and then they exert a torque on each other. And that causes it to rotate. And not only, what's really fascinating is not only do the pieces rotate, but the individual gel also rotates about its center of mass. So there's the global motion of the 4, and then there's rotation of the individual gels around their center of mass. Now we think we understand this sufficiently well that we can control this behavior. So we can control the chirality of the motion of this pinwheel. Furthermore, so it's the luck of the draw. It's the random number generator. So if we take a random number generator and it's such that we do get this rotation, then if we increase the box height, then we can stop the rotation. I'll explain that in a minute. If we have a random number generator such that it's not rotating, then we can introduce activator, the U, at the boundaries or in the middle of the box, and we can get the rotation. So what this is saying is that you need a critical concentration of activator to induce this rotary pinwheel-like behavior. And what I'm doing here is by increasing the box height, I'm diluting the amount of U in the system, and that's arresting the rotation behavior. So you need a local critical amount of U of the activator to get the rotation. So if I get this rotation, then 50% of the time it'll be, or 49 point something, rotating in one direction, and 50% of the time, or 51% of the time, it'll be rotating in the other direction. So it's arbitrary. But I can paint the sides of the box now so that if I do get the rotation state, then I can control the chirality of the motion. So if I paint the box with U being highest in a clockwise direction, then I will get clockwise rotation. If I paint the side, these are gradients of U. Red is highest, and this blue is lowest. So if I just paint the sides of my simulation box, then, in counterclockwise direction, then it'll move in a counterclockwise direction. And I was just pointing at, this is counterclockwise and that's clockwise. So paint clockwise, it moves clockwise. Paint counterclockwise, it moves counterclockwise. But the scenario is a little more complicated than that. What's really amazing is I told you that if I take one luck of the draw random number generator and it didn't oscillate, but I introduce U, I just need to introduce that U at an initial stage. So I introduce a high concentration of the activator at some early time. Here I put it at the sides of my simulation box. And then I can wash it away. But it has a memory. That high concentration of U somehow has already set up the scenario such that it will oscillate and rotate. Right? So the one that didn't rotate, for a finite amount of time, I introduce a high concentration of U. I wash that U away, and it drives it to rotate. And a similar effect, the story is similar. I paint the sides of the box for a finite amount of time. I wash it away, and I can control the chirality. I see Len standing up. I have one other. Oh, I have about 3 more slides. So, is that all right? 3 more slides. And they're sort of conclusion slides. Is if I take 16 of them, then they can form these little rotating things, and now it looks reminiscent of Igor's beautiful picture of these multiple gears. So perhaps in the future we can make more complicated self-assembled machines, not just these gears. And I'll finish with one last one. And this was the temperature controlling scenario. And it's a homeostatic device. I'll just quickly tell you, and this we did with collaboration with Joanna Eisenberg, that we made an oscillatory system where none of the components oscillate, but it's just a beautiful system. She has these fibers. She has a binary fluid that she maintains through a microfluidic device. She has catalyst on top of these posts. She starts with the posts upright. They interact with the reagents on top. That liberates heat. Because it's an LCST gel, the gel shrinks. It tips the post away. The heat dissipates, the gel expands, and the posts are driven back up. So it just keeps oscillating, and through those oscillations, it maintains the temperature of the system. And with that, I'll conclude, and I thank you very much for your attention. Oh, thank you. My mom's really happy you said that. Oh, thank you.
Have you tried with no symmetric distribution of the cubes or with like higher number of cubes?
Oh, we did, yeah. And you get amazing behavior that we just can't explain yet. You do get, like Igor has this gorgeous movie where he has these creatures attacking other creatures, and we have that too, that they do exhibit some sort of swarming behavior. And the ones that aren't rotating will attack the ones that rotate. And so there's very, very complicated behavior. And we're in the process of trying to understand that.
So how small can you make your gels? Can you make them at the micron scale?
Oh, yeah. So I should have pointed, thank you so much for asking that. When they're 0.5 to 1 micron, so when they're small, they're not interesting. I mean, they're still interesting because the chemistry and the mechanics are slaved. It just, they're slave to the chemical oscillations. The mechanical oscillations are in time to the chemical oscillations. When they're larger is when you get a traveling chemical wave that causes all sorts of complicated behavior. So small to us is not so interesting.
But does the size change if they're small?
Yes, oh yes, of course it does.
Get them to go, what ratio?
Well, these are relatively delicate, fragile gels, so they're about 10%. But Ralph Nuzzo has just made a very beautiful BZ gel, and where it's about 50%. And that's actually published.
Another question about the pinwheel. So what actually keeps them apart, the different pieces?
That's a really good question. So artifact. So I told you that I introduced an excluded volume interaction between them to keep them apart. When I showed this to Ryo Yoshida, he said that's not a problem. He can functionalize the gel pieces. He can put little hairs on the sides of the gel pieces to have steric repulsion. And those polymer chains can also be BZ gel, BZ chains. He's actually made BZ hairs. So Ryo could do that. And we're in the process of working with him to do that. Yeah.
How important is the friction with the floor?
How it works with the... Well, let me just answer that. How it works with the friction with the floor is that I haven't included it in the model. But again, experimentally, that's not such a problem because you can functionalize the surface, the glass, so that it'll have very little friction, minimal friction as it moves. Yeah.
So what's the power return of your photoresponsive gels? Can it be used for pumping, like running mucus, peristaltic pumping?
Well, yes, but think of, from a manufacturing point of view, think of this amazing power where you start with one piece of material and you can shape it into any structure that you want. And then you can do more photoradiation to enhance the cross-link density so it stays in that shape, for example. Or you shape it into one structure, use it for a finite time, then take the light off, and then let it be its original shape, and then shine light through a different aperture, and you get a different shape. So that's the... Yeah, I mean, the walking is quite lovely. It's very slow. You might be able to use that for transport in a microfluidic device, for example. No, it could do. I can't. I mean, it's not mucus. I don't know where the mucus comes from, but I'm not sure about the mucus story. But you know, it could on the tenths of microns per second, it could be used for transport in a microfluidic device, and that you could direct it with light in a microfluidic device, so in a very non-invasive way. That's the real power of the use of light.
Well, thank you again.